absolutely pleased to be here to speak with you today about the development of breast cancer vaccines, and particularly what we're focusing on in Artemis. Before I get going, though, I should disclose to you that some of the vaccine approaches I've been working on in this area for probably, I don't know, I guess I lost count, about 15 years now, and some of the approaches, many of the approaches that you will hear from, for example, you've heard from Brian Zernicki and Dr. Mittendorf, and I'll be covering some vaccine strategies I've been involved with since the mid-1990s, I guess, and in some cases I've turned over in all cases I've turned over all my rights to the profits from those to the companies that I work with. But I should point out to you that the academic institutions that I work with still retain, some capability of collecting on that. So I just wanted to put that out forward. My commitment here is for personal reasons. I have a family history of breast cancer, and my commitment is to you. So let's talk a little bit about the Artemis project. And I hear a bit of an echo. I don't know if that's if you can. Just me? It's my ears. Okay. So yeah, coming with age. So certainly. So let's look over what I'm going to talk about, an overview of the Artemis project, and I think that Dr. Ewald gave us a nice overview of what's going on. Most of the vaccine research that we're focusing on now is the tumor-associated antigens, and I'll get into what that is in the secondary prevention setting. And I'm going to talk about what vaccines do so that you have an understanding about why we would target a tumor-associated antigen versus a viral antigen, et cetera. The vaccine approaches that are being considered. I'm going to move fast through some of the ones that Dr. Ewald spoke of. And this idea that we need to approach safety very meticulously because of the current public perceptions of vaccine, and I'll talk a little bit about that in the case of Dr. Wakefield's publication back in 1998 regarding the link between the MMR vaccine and autism. And the most important component of this is that I think, and hopefully I can, through discussion, help you think that Artemis really embodies a realizable goal. So what we know from the murine modeling that we've done, as well as a variety of different human strategies, vaccines have the potential to prevent breast cancer, whether it's a viral cause or other causes that are undefined. And so the idea is not to nitpick through this diagram that was drawn up nicely in the Artemis documents, but kind of extend to you this idea that the Artemis project embodies this complex pathway-driven approach so that we actually can develop an outcome here on developing a vaccine. And we're targeting 3 different areas. You've already heard about the viral approaches, so we need to understand if there is a bacterial or a viral cause to breast cancer. And we also need to understand whether or not we can target what we call neoantigens. So those are new antigens that pop up in breast cancer that potentially we could harness with respect to vaccinations. And then talk about, in a little bit more detail, because where all of the work is being done is on targeting normal self-antigens that are overexpressed in the tumor microenvironment. So I'm not going to go through the vaccine history, but there's a long-standing history about vaccines. We know a lot about what vaccines do. We know how they act. It's been something that we've known about since 430 BC, as you can see on the slide there. The idea that we could capture the essence of the immune system with respect to the development of cancer came from Dr. Paul Ehrlich, a 1908 Nobel Prize winner in medicine, that established this idea of tumor immune surveillance and the idea that we could intervene with a vaccine. But it's only within the last several years that we've actually developed the molecular tools, the molecular concepts to move forward into a rational design of vaccines. And so before 1970, for example, many of our vaccine strategies focused on the development of vaccines based on attenuated viruses, attenuated bacteria, heat-killed bacteria, viruses, etc. The newer vaccines that we're seeing now coming into the clinic are molecular based. So we can actually synthesize these things. There's no organisms involved or anything, and they're very effective. Cervical cancer vaccine is certainly one of those. How do vaccines work? Vaccines work by eliciting a component of the immune system called the adaptive immune response, which consists of multiple cell types. It results in long-lasting protection against infectious diseases. We think, based on our animal modeling studies, that can result in long-lasting protection from cancer as well. This is just a diagram of a mixture of cells that you would find in your blood. The red cells, of course, are the red blood cells that carry oxygen throughout the body, and a complex mixture of white blood cells, which are colored in various colors here, which constitute the B cell and T cell components. So this is very important because B cells would make antibodies that would recognize whole proteins that are presented by the tumor or other tissues in your body. Whereas we now know that T cells recognize only fragments. And this is very important because tumors tend to upregulate proteins and present new fragments of antigens that your immune system wouldn't normally see. So these represent novel targets that aren't being presented by MHC class I molecules in the rest of your body. And MHC class I molecules are those transplant antigens that are present on all cells. And essentially what they do is they hold up fragments of proteins that the immune system can see, and specifically T cells. We're specifically interested in the CD4 T cell subset, which is what we call a helper T cell subset, which drives a very strong memory response, as well as the CD8 T cell response, which are directly cytotoxic to cells that are infected with viruses or cells that are malignant or pre-malignant as well. So we know that there are 27 diseases that are now vaccine preventable. This shows us that the immune system is very complex. In fact, it's one of the only tissues in your body that has memory, in fact, besides the brain. In fact, the memory the potential for memory in the immune system matches that of the brain, no doubt. Your body can create all kinds of T cells, millions and millions of different kinds of T cells that are genetically different than the rest of your body that can recognize these novel targets that are on cancer cells or viral infected cells. So I left a little spot down on the bottom there on the right-hand side, and hopefully, you know what I mean, right? Okay. So okay, so I'm going to breeze through the viral antigens because you've already heard about that, and I think Dr. Ewald did a good job. We have very little information. There's been a few studies that have looked at the role of viruses or bacteria in the development of breast cancer. We know they're fundamentally important in liver cancer and in cervical cancer. In fact, the new vaccines for cervical cancer not only can prevent the infection, they can recognize the damaged pre-malignant tissues that develop in your body and kill those directly without ever seeing the virus. So we know now that the immune system, based on that data, can actually find those cells in your body and kill those, and we think it can be done for breast cancer as well. We didn't know that cervical cancer was caused by a virus until 1976. We just got the virus. The dawn of the molecular area of immunology was just yesterday. We're in a really important position now to capitalize over the next few years at developing vaccines, whether or not they're based on self-antigens or viral antigens. So this is just an example of some of the new technologies that are coming available. This is from the Lawrence Livermore Laboratory. We now can detect bacterial pathogens or viral pathogens of all different types. This particular one detects about 3,000 different microbes. We need to apply those technologies to breast cancer to try to understand what's causing breast cancer from a microbial standpoint. There's been a smattering of studies that have been done, very limited, looking at EBV, as you've already heard, mammary tumor virus, as well as, 5 minutes, okay, as well as Epstein-Barr virus. And so we need to move in a very focused approach, looking at a wide variety of other causes in addition to whether or not these viruses, because it's still unanswered, cause breast cancer. There's neoantigens. What that means is that the tumors themselves actually can make new proteins. And so this is just a reminder in case you've forgotten this or never knew this. The kind of paradigm of life here is that you encode proteins within your DNA. There's a messenger called RNA, which is a molecule that helps the body synthesize new proteins. You're doing this as we speak now, copying DNA into RNA, moving that into protein, making new proteins. And what we know is that your DNA makes 23,000 different proteins, or we think it makes 23,000 different proteins. And not all of those are expressed at very high levels in your body. And they could contribute, some of those could contribute to breast cancer. So what happens when breast cancer starts dividing uncontrollably or even pre-malignant lesions is you lose this integrity of being able to copy your DNA, and as a result, you get new DNA that results in new proteins that the immune system has never seen. Some of those may be important to the growth of the protein. We don't know. One of the studies that was just published at the Mayo Clinic in 24 early primary breast cancers shows that there's lots of new proteins that are being synthesized by tumors that are potential targets for vaccine. Now, we don't understand the role of these in causing cancer, but what we know is that these new proteins show up early in breast cancer, and so it's worth considering a lot of these proteins in the development of breast cancer vaccines. So the last one, and where most of the work has been done, is with the development of vaccines targeting what we call tumor-associated antigens. So these are antigens that are normally present in your body. And you're going to hear a lot about this at the workshop today, and I forget exactly what time that is, but it's the vaccine workshop if you're interested. I'm going to fly through this. So we worked several years ago on developing a vaccine for HER2/neu, and Dr. Cernicki's work shows that this is potentially effective at secondary prevention and possibly even primary prevention. This is a cell-based approach, so it's a little bit difficult thinking ahead in terms of primary prevention, but the fact is we can target normal proteins that are in the body, and because of this amplification, they present new antigens. And in this particular case, he takes these HER2/neu antigens that are overexpressed in the body, pulses them onto a special subset of T cells. That results in the generation of immunity. He has very nice data, and it's actually, I'm going to skip that slide because it's just redundant with the last slide, but he has very nice data, and it was very reassuring to see this because this is some of the epitopes that we had, or antigens that we had worked on in the 1990s. And what he's seen, at least in some subsets of patients with DCIS, he can actually induce, prior to surgery, complete clinical regressions of those lesions in these patients, so that when they go to surgery to remove those lesions, nothing has shown up. This results in the generation of immunologic memory as well. So the hope is that the patient will have lasting immune memory. We may need to boost from time to time that can prevent the recurrence of the tumor. And we've been looking at continued identification of epitopes from HER2/neu and other tumor antigens that I'll talk about. when breast cancer develops, your immune system responds to it naturally. It escapes it for reasons that are unclear, but we can use that signature or those signatures to help us develop new target antigens for HER2/neu. And this is just an example of some, if you look on the diagram on the left-hand side there, regions where your immune system is responding to. So we're excited about this. In fact, I'll discuss some of the new clinical trials that we're engaging in, looking at the development of immune responses to these regions to see if they are potential for prevention of cancer. So Beth Mittendorf is going to talk about the new vaccine, which is very closely related to this in phase 3 clinical trial. And I'm sorry for such a graphic, and I'll just give me another minute here, but there is a lot of noise that has been made in the past few years from Dr. Wakefield's study that was published in Lancet, subsequently retracted, showing a connection between MMR. Very carefully crafted article that wasn't true. And a company was built up around identifying biomarkers of autism in patients or in children being immunized with the vaccine. So there's no evidence. There's no evidence whatsoever. But despite that, if you look on these charts here, you can see several lines down that 25% of individuals still think that vaccines still cause autism. That's just not true. Vaccines are very safe. We've immunized over 150 patients, and I have to tell you, the adverse events are extremely minor. I hate to even call them adverse events because it's like a little skin rash in some cases, or maybe just a transient feeling of the flu or something like that eventually goes away. Vaccines work. They prevent disease. This is contrary to a lot of public perception because of the Wakefield study. So, and this is just an example of the pre-vaccine cases of various diseases and in 2008. So with that said, Artemis is really positioning us to move forward with what I think is an outstanding program to get to the point where we have multiple options for breast cancer prevention through the use of vaccines. And I think it's a realizable goal because of the technology that we have today, that within the next few years, we can be testing this in clinical trials. So with that said, I just want to thank everybody for your attention and their...
Thank you.